Okay, so yesterday we talked about the cell theory, okay, and just quick review here. First point of the cell theory is that all things are made up of cells. Not all things, all organisms, living organisms are made up of cells, okay. The second point we said was the most important, and that tells us what the role of the cell is, and the role of the cell is to carry out the basic functions of the organism, okay. Basic functions would include respiration of some kind, okay? So that would be the exchange of oxygen and carbon dioxide, or if you're a plant, the other way around, carbon dioxide for oxygen. If we're talking about digestion, acquisition of energy, okay, or photosynthesis, if you're a plant, the excretion of wastes, okay, how those are rid by that particular cell, okay, reproduction of the cell would also be a basic function, and then of course the third point of the cell theory was about how cells do in fact reproduce, okay, they reproduce by splitting in half, right, it's called binary fission, and that's the only way that cells are produced, all right. Everybody with me there? Okay. So handling techniques for the microscope. Okay, so these are our microscopes. You can see already on this one that their design is not perfect. All right, in that they have this incredibly large, heavy adapter converter thing that converts AC to DC. Okay, but it's way bigger than the gauge of the wire used to connect it to the microscope. The inherent problem there is that they always break the wire. That's why it's covered in electrician's tape. Okay, it's like all MacGyvered up. Okay, so we've had to do that on several of the microscopes. So we do have to be very careful with the cord. All right, and especially here where it goes into the microscope, it is especially vulnerable to breakage there. Okay, the big thing is that always wrap it around the microscope when you are done or when you are moving around with the microscope. Okay, it should never be hanging down, because if it does, okay, you will notice that you can step on it quite easily, and then as you bring the microscope forward, tension begins to appear in the cord, and one of two things, or both, will happen. You will rip the cord out of the microscope, or you will drop the microscope, or both, and that's not good, okay, because then you'll hear the crashing sound. Then you'll hear the thumping sound, then the ambulance sound, and then nothing. Okay, so we don't want to have that happen. Okay, so please treat them very carefully when you're carrying them around. When you carry them, they are carried with one hand on the base and one hand on the arm. Incidentally, this is the arm. Someone teaches that it's the neck. I would never hold someone's neck like this, okay? It's just weird, right? It's an arm, right? Just gently hold the arm, okay? That's how you carry the microscope. Two hands, always two hands, right? Because I've seen this happen. Someone's carrying it around like it's a bowling ball or something, and someone comes up behind them and gets them right here on the funny bone. What's the natural reaction of your hand? It lets go. Okay, and the microscope goes crashing to the ground. Always with two hands, okay, that way if you get bumped or jostled, the microscope doesn't fall to the ground. Everybody with me there? Okay, so that's handling techniques, okay, carrying techniques for the microscope. Now, use of the microscope, okay, involves a lot of dials and knobs. Okay, so we'll quickly look at the diagram of the microscope here so that when I'm using terms, you kind of understand what I'm talking about. Okay, so you can see here, right, I don't think you have this diagram in your notes, guys, because I just put it on. All right, this part here is the arm. Okay, we just said that a minute ago. Okay, this is the base. All right, it has rubber feet on it. Okay, ours do too, because that's exactly the same model of microscope. Okay, you'll see that there are two focusing knobs on the microscope. One is very big and one is much smaller. Okay, this is the coarse focus knob. When you turn it, okay, and so this one here is the one we're talking about right now. When you turn it, it moves the stage, that's this flat part, okay, it moves the stage a lot. In fact, you can't turn the coarse focus knob one complete turn before it hits the top or the bottom of the allowable movement of the stage, right? So very small movements of that move the stage a great deal. The fine focus knob, you can spin and spin and spin and spin and spin, and it almost never stops, okay? It moves the stage very, very little. In other words, it allows you to very finely focus something, whereas the coarse adjustment knob is very quick, right? So the coarse adjustment knob, you're really only ever going to use when you're using the scanning power lens, and that is the smallest of the three lenses that is on ours, and it has a little red band on it. Okay, so this really short one that I'm pointing at right here, okay, that one there is the scanning power lens. It's the one you use first to just find things, okay, to scan the slide that you're looking at, find the specimen, get it centered and focused, and then move up to a higher magnification. All right, so you'll probably only use the coarse focus knob on that. You might use it once in a while on the low power lens. That's the second one, okay, it has a yellow band on it. You might use it once in a while there, but you will never, I repeat, never use the coarse focus knob with the high power lens. The reason being is the high power lens is very long, and as a result, it can touch the slides, and the coarse focus knob has a lot of torque. So if you are twisting on the coarse focus knob, you can actually drive the slide up into the high power lens if someone has made adjustments to the stage. There's supposed to be a little screw in there that stops you from doing that, but some people think it's funny to adjust that, and once in a while it's been adjusted so that you can actually turn the coarse focus knob far enough to smash the slide against the high power lens, which breaks both the slide and scratches the lens. And again, then you hear the thumping sound, the ambulance sound. Okay, so we don't want to do that. So we don't use the coarse focus knob. The other part of using the coarse focus knob is it's useless on the high power lens anyway. You go right past focused so fast that you never even know you passed it. All right, just because it's such a higher magnification, and it's really hard to use the coarse focus knob for that. So that's why you use the fine one on high power only. Okay. All right, other parts of the microscope. Okay, we talked about the stage here. They've got, this is a slightly different model than we have, more like the ones you had at John Paul II, okay, that have the stage clips like this. Our stage clip is much better because we have a movable stage. Okay, our stage can be moved around using these knobs here, which is really good if you're trying to find something on the slide. You don't have to put your finger on the slide and move the slide around. The problem with that is when you do that, you usually push the slide down and then it goes out of focus. With these, you don't have that problem. So when you clip a slide in, okay, you move this little catch out of the way, put the slide in there and then release it, and it'll snug up and hold the slide in place. And then when you're looking through this, obviously not like that, like this, okay, you just move this around to move the specimen around in the field of view. Okay, everyone follow me there? So those parts aren't really on that particular, they're not on this diagram here. There's the two little knobs that move down there. Okay, so yeah, they're here and there, and yeah, I don't know, I can't draw. Okay, so then we've got this is the nose piece here, the head or the nose piece, right? And that's where, of course, the objective lenses are, high, low, and scanning power. Okay, we got our eyepiece tube or ocular tube, it's sometimes called, and our eyepiece, that's our last lens or our ocular lens because it's the one that's right next to your eye. Okay, it magnifies an additional 10 times. That's usually the one you need to clean. Okay, if you're seeing stuff and it's not, when you move the slide, the stuff doesn't move with the slide, then it's something that's on the lens, and it's usually on this lens because that's the one that's closest to people's face. Things like eyelashes and stuff fall on there. If people are wearing mascara, I find that that's the worst thing, okay, because it comes off in little black clumps, and it actually is quite greasy, and it sticks to this, and then it smears on there. Obviously, you're probably still going to do your makeup and everything on a day when we have a lab, but just know that there's two problems with that. First off, obviously, it makes the lenses dirty, but it also has a tendency for you to see your eyelashes a lot more when you're trying to look through the lens, and it can be very distracting and actually difficult to use the microscope if you've really gone crazy with the extended lashes stuff. Okay, so yeah, and don't wear the fake eyelashes on a day when we do a lab. I actually had a kid who had those once. They were like up to here. Okay, it was crazy. But you cannot use a microscope with those because you can actually hear them clicking against the microscope every time you blink. Okay, so you want to avoid stuff like that. If you wear contact lenses and glasses, obviously you probably have both. I always find it's easier to use the microscope if I'm wearing my contacts. just because I can get closer to the lens. I don't have to worry about banging the lens on my glasses, things like that. I find anyway, I tend to see better if I'm wearing my contacts. So tomorrow, if we're going to be in the lab, if you wear glasses but you have contacts, it might be easier to wear your contacts. I mean, you can see for yourself. But all right, other things here that we haven't talked about. Oh, the lights. Okay, the light source is down here at the bottom. Okay, it's just a little LED lamp. It's got a little power. It's got a little switch on the back here, and it has a rheostat that allows you to adjust its brightness. Now, one last thing that's on the bottom of the stage. They don't show it very well here because of the angle the picture's taken at, but there's this condenser lens that is on the bottom underneath here. Okay, it's this little apparatus here. Okay, and it's got a little lever on it. Essentially, it's an iris, so it opens and closes. So you've got the ability to brighten and dim the light, but you also have the ability to let more or less of it in with this little adjustment. That's really useful for not this lab, but our next lab, where we'll be looking at unstained cheek cells, okay, from your own mouth, okay, that will smear onto a slide. You'll be looking at those. They're much easier to see if you dim the light and increase the contrast with that. You won't need to adjust that tomorrow, most likely. Okay. All right. We can't look at blood or anything like that because there's, hepatitis and HIV and stuff, so we're not allowed to do that anymore. Okay. Back in the day, we were allowed to do that. You just pricked your finger and looked at your blood and whatever. But, okay. All right. So those are essentially the parts. So now if I go back again to handling techniques, it'll make a little more sense when I'm talking about them. Okay, so they're always carried with one hand on the arm, one hand on the base. They're always returned with the lowest objective lens in viewing position. Okay, if you return them like this, and people are used to having the short lens in position, they'll grab the coarse focus knob, crank away, and smash. All right, so you always, when you're done, return it to the scanning power lens, right? Then it reduces the chance of damage, but it's also the one the next person is going to want to need first, going to want to use first, or need first. Okay, so everyone with me there? Always return it with the scanning power lens in place. Okay, always obviously start then with the lowest power and move up. The reason you do that is because of the field of view. When you look through the scanning power lens, you see a big white circle. Actually, you see a big white circle no matter which lens you're looking through. Okay, but on the scanning power lens, that big white circle is really big. When you look through the low power lens, you magnify more, so you see a smaller area. Okay, it's like if you were, let's say you went to watch a football game and you were looking at the other side of the stadium. Okay, you look at the other side of the stadium, you can see pretty much the whole stadium, right? The whole other side. All right, now you put a pair of binoculars in front of your face. How much of the other side can you see now? A lot less, right? Like your field of view has gone from almost 180 degrees down to like 15 degrees maybe. All right, now what you see through the binoculars is greatly enlarged. You can probably see people's faces, all right, and things like that, right? So you can see individual people, but you can't see nearly as many people as you saw before because now you've magnified a smaller area. Okay, everyone with me there? When you go to high power, it's even worse. Okay, high power would be like if you took a telescope and looked at the other side of the field. You probably wouldn't be able to find anybody, but if you happen to be on somebody's shirt, you could probably read the tag, okay, because you had a high-powered telescope. So you're looking at an area like that. But what you see in that tiny little area is greatly magnified, right? So we always start with the scanning power because we can see more. We can't see more we see much less detail, but we can see more area, and that allows us to find things more easily. All right. So if I go back to the football game example, if I knew my buddy was wearing a bright yellow shirt, okay, and I wanted to find where he was on the other side of the field, I would not use the binoculars to find him. Okay, I would first look over until I spotted yellow shirts. There's probably going to be several. All right, and then I would take out my binoculars and look where those yellow shirts are. That's not him. That's not him. Okay, until I found the right guy. But I wouldn't take out a high-powered telescope and try and scan the other side of the field for one person. Okay, so we always start with the greatest area first, and we move down. Okay, move up in magnification and obviously down in viewing size. All right, and then use only lens paper to clean the lenses. We talked about that yesterday. There's a real tendency for people to just, out of instinct, use their finger or their shirt, okay, or whatever. Well, even some synthetic fibers are capable of scratching some of this stuff. Plus, you just don't know what kind of grit may have been on your shirt or whatever, even on your fingers as well. It's microscopic, right? And you don't feel it, you don't notice that it's there, right? Don't use Kleenexes either, because they leave little tiny pieces of lint and stuff on there, and when you look through the microscope, it's distracting. So there's special paper that'll be on the front table. If you feel like your microscope needs to be cleaned, you come up, you grab one of those sheets of paper, and you use one part of that paper on this lens, and do each of the other 3 lenses with a different piece of the paper. Never use the same piece of that piece of paper, right? So you got like 4 corners, use 1 corner for each lens, and then throw that piece of paper away. Okay, everybody with me? All right, so this is also not in your notes. You're going to need to draw it. This is the field of view for each of the lenses that we're going to use, and it's going to help us to figure out how big the things we're looking at are. Okay, it helps us with our estimation of size. Okay, so when I look through the scanning power lens, the scanning power lens magnifies what I look at by 40 times. So whatever I look at is 40 times larger, okay, than it appears normally. The circle that I can see, which we call our field of view, or FOV, okay, field of view, is 4,000 micrometers. You're probably thinking, what the heck is a micrometer? Okay, well, a micrometer is 10 to the minus 6 meters. You're probably thinking, okay, that really didn't help me very much. I have no idea what 10 to the minus 6 is. Okay, well, give you a better idea, this is 4 millimeters. 4,000 micrometers is 4 millimeters. All right, a millimeter is how much of a meter? 1,000th. Okay, a millimeter is 1,000th of a meter. Okay, a micrometer is 1,000 times smaller than that. Okay, so very, very small. So let's say I'm looking at something on my scanning power lens, and it's about yay big. How big is it besides yay big? Just an estimate. How big? All right. I'd say that's a pretty good estimate. Okay, it's not quite half, but it's not quite a quarter. Right? Somewhere between a half and a quarter, about a third. So let's say it's about 1,500 micrometers. That's how we estimate the size of things. Okay, if we know how big our field of view is, then we can calculate. Well, that's missing. Sorry, guys. Didn't even notice that. Okay, this one is 1,600 micrometers. Okay, if we know how big the field of view is, the diameter, then we can estimate how big the object we're looking at is. Okay, and that's what we did here. We said it was about 1,500. Right? And I'd say that's a pretty reasonable estimate. Now, like we said before, if I take this object and I'm looking at it on the scanning power, you know, it's this big. It takes up that much of the field of view. When I look at it on this one, it's going to fill up the field of view. If I look at it on here, I'm only going to see little itty-bitty parts of it. Is that the idea? It is. Okay, that's kind of the idea here. The stuff we're going to look at tomorrow, like the amoeba and the paramecium, when you look at them on scanning power lens, they're actually fairly large for a microscopic organism. They're several hundred micrometers. When you look at them on scanning power, you'll see their shape and maybe a little bit of detail, but not much. When you get them on low power, you'll be able to see quite a bit. Right? When you get the amoeba on high power, you can't even see the whole thing anymore. Right? You're looking at different parts of it. You can't even focus it all at one time. You actually have to focus, because it's a three-dimensional object, you actually have to focus on the front of it and then focus a little more to see a little deeper into it, focus a little more to see a little deeper into it, okay, until you see kind of the whole thing, right? The high power lens is almost overpowering for something like an amoeba, right? You guys follow what I mean by focusing through it? Okay, it's like right now, if you're looking at me, the person in front of you is not focused, okay? Like right now, I'm looking at Dylan, and Emily is not focused, okay? Whereas if I look at Emily, now Danielle and Dylan are out of focus, and I don't see their detail, right? So when I'm looking at that amoeba, okay, Emily would be the closest part of it to me, and then I would, if I wanted to see past her, I would focus till I was on Danielle, and then focus until I was on Dylan, okay? So that makes sense? Okay. All right, so does everyone get the size estimation part? Okay. It's really, a lot of people really get confused with it, but it's quite straightforward. If it takes up half of the field of view, and the field of view is 400 micrometers, how big is what you're looking at? 200, yeah, 200 micrometers. If it's a 400, sorry, I said 400, yeah. So if it's 400 micrometers and it takes up half, it's 200 micrometers. It's not a difficult thing, and it doesn't have to be exact, okay? If you tell me that your paramecium was 182.479 micrometers, I'm going to call you on that. There's absolutely no way you know that, okay? You didn't estimate that. You just estimate. It's like, oh, well, it took up about half. Maybe a little less than half. It was 200. It was 180 maybe. It was a little less than half, okay? You just have to estimate. Does that make sense? Okay? I'm not looking for exact numbers. Now, looking at this object here, okay, we naturally estimated it. It's not symmetrical, right? We naturally took which dimension, shortest or longest, when we did our estimate? Longest. And that's what we generally do. If we're looking at an object that's got a definite longer dimension, you always measure on the longest dimension. Okay? If it's fairly symmetrical or circular or something like that, or its shape is so random that you can't really tell, then you just kind of go in general. But if it has a definite long axis and a short axis, you go along its longest axis. Okay, so that's all of this stuff here. Okay, we got this, the lens diameters, we've got the estimating the size, we've talked about that, so we're good there. Okay, and we kind of reviewed that already this morning. Okay, the 3 points of the cell theory we talked about. Who are the 4 people? Anyone remember from yesterday? Hooke, he discovered cells. Lee's trying to pronounce it. That's not bad. She even got the V in there. Leeuwenhoek. Yeah, okay. You don't have to remember their first names. Their last names are good. And Schleiden and Schwann, okay, the 2 Swedish guys who actually came up with the cell theory. All right, and then the third point, okay, on what basis can we deduce that plants and animals may have had a common ancestor? Okay, this we didn't get to talk about quite as much, but we did that little comparison where we counted the number of things that were similar and then the number of things that were different. And they had way more things that were similar, okay? And that doesn't happen by accident. You look like your mom and dad because you shared genetics with them, okay? Well, all eukaryotic organisms, that's organisms with complex cells, have the same kind of cellular structures because at some point they shared a common ancestor. All right, we trace that kind of evolutionary link in history back a long way. Right? If you look at all mammals, for example, and you look at their 4 limbs, okay, for us that's hands, right? But if you look at the bone structure in the 4 limbs of all mammals, it is remarkably similar, even if on the outside they don't look anything alike. The bones in your hand look like the bones in the wings of a bat, the bones in the fin of a whale, okay? Even to some extent the bones in the paws of your dog or your cat, okay? All of those, they're all similar because we shared a common ancestor somewhere along the way that had front limbs, okay? That had carpals, okay, carpal bones and things like that are all part of the hands, okay? All right. Okay. Lab diagrams, okay? Something that we haven't talked about yet, but need to, okay? Drawing a proper lab diagram, and you know what, I probably should have photocopied this. I will for you. There's going to be 2 ways that you can go about doing that, okay? There's the way that universities always wanted before, but I would imagine now that they've probably changed with the advent of digital photography. It's a lot easier to do stuff with that now. So we've been looking at doing 2 things when we do diagrams for lab reports. The first thing you're always going to do is take a picture of what you're looking at in the microscope, okay? And you can do that with just a little bit of practice. you get it focused in the microscope, and then stick your phone's camera lens right on the eyepiece and just move it around until you've got what you were looking at on the screen. It takes a little while, you got to get the angle right, but you can eventually get it so that you can take a very good picture of what you were looking at through the microscope with your phone. All right, that's always a good thing, because then when you're going back to do the lab drawing, you can either look at the picture you took and do a drawing, or you can take that picture and insert it into your lab report and put the labels on it from there. Okay, I'm fine either way. All right, if you would rather draw it by hand, I'm okay with that. Okay, if you draw it by hand though, you have to take a picture of it and insert that into your lab report, so you're doing it one way or the other. Candy. No, you'll take it on whichever one gives you the best view. Most of the time that's high power. There'll be exceptions to that. Tomorrow we're going to do a wet mount of the letter E, okay, from a newspaper, and you can't look at that on anything other than scanning power, or you don't see the whole E. Okay, so yeah, they'll do the scanning power for the letter E, and then pretty much everything else will be high power. You might use low power for the amoeba, depending on how big of an amoeba you're looking at. Okay, but whichever one seems to give you the best view of what you're looking at is the most logical choice to take the picture on. Okay, so whichever way you do this part, the actual picture of the organism, whether you're doing that with a digital picture that you've taken or whether you're going to draw it by hand, everything else format-wise needs to be the same. Okay, so format-wise, your name and the date go in the top right-hand corner, always. Okay, they always go in the top right-hand corner. Your labels are always made with horizontal lines, and they all go to the same side. Okay, we don't want them going haphazardly everywhere, because it makes the diagram look busy and crowded. Okay, so we want them all going to one side. Okay, and they're all horizontal lines. Now, that can become a little bit of a problem if, let's say, I needed to label this thing and I needed to label this thing that's right behind it. Now, how do I draw two horizontal lines without them overlapping? Well, in that case, you draw up and over. Okay, you always end your label with a horizontal line. Okay. And then at the bottom of the diagram is the title. That's the part everyone screws up, because your whole life and in everything else in life, the title goes at the top. Well, on lab drawings, just to be difficult, biologists want it on the bottom. Okay, so the title goes right underneath, and then you tell me the magnification at which you viewed the organism. Okay, so if it was scanning power, it's 40x. If it was low power, it's 100x. If it's high power, it's 400x. All right, everybody with me there? Okay, so you just write the magnification that you viewed it on, and then you give me the size as you estimate it to be. Right? All of those things need to be included on a lab drawing. So name and date, labels, all to one side, title, magnification, and size. I'll give you a copy of this so that you've got it. Okay, I should have copied it, but I'll get it to you today. All right, is that making sense to everyone? Okay, lab drawings they're fairly straightforward, and I would say probably for the most part you're probably just going to want to use the picture you take with your camera, unless you really like drawing. Right? For me, like the advent of digital cameras was, I don't have to draw anymore. Okay. All right, so that's what we know for that. Okay, so we'll leave it at that, and then we'll start talking about the lab here. We'll do that separate.